My name is David Koslicki. I'm an associate professor of computer science and engineering, biology, and life sciences at Penn State University. And today I'm going to be giving a talk about a statistical test, a simple statistical test, to test for presence and absence of a genome within a metagenome. This microphone I thought was way too hot and loud, so I moved it like way down here. So if the people in the back or whatever can't hear me, please do let me know. Awesome. Feel free to ask questions during the presentation. No need to hold them to the end. I know that these are oftentimes recorded, but if you have a question, I'm sure somebody else who watches the recording would also have that question. If you're curious, all the graphs in the background of this intro slide are different De Broglie graphs for different k-mer sizes of a microbial community. Okay, so what am I going to talk about today? So I'm going to be talking about community membership hypothesis testing, basically taking a metagenome and trying to see whether a reference genome is in it or not in it. So after some motivation, we'll talk about the algorithm, which we call YACHT, talk about some experiments, and then conclude with the code and future work. Okay, so first up, metagenomics. I'm aware that not everybody here does metagenomics, so a one-slide quick introduction of what it is. It's a study of microbial communities through their sampled DNA. Okay, sounds straightforward enough. Why should you care? So microbes are absolutely everywhere. They're ubiquitous. They're on basically every surface on the soil, in and on you. And in fact, there's basically as much bacterial cells as human cells in and on your body. So whether you're human basically or microbial, it's kind of a toss-up. And even worse than that, there are 100-fold more bacterial genes in and on you than human genes. So as you might imagine, that means that microbial communities are implicated for lots of diseases, but also some extremely life-critical processes. And the reason why this can happen, where we have so many bacterial cells in and on us, is that the microbial cells are very small in comparison to human cell sizes. So this big orange piece right here, that's a white blood cell, and around all these bunch of clump of green little guys are bacteria. Okay, so that's why you should care. What is the data? What do we use to study it? So first off, you have to go out and obtain a sample. And what I'm going to be talking about here is whole genome shotgun metagenomic data, so not amplicon and 16S kind of thing. So you go out, you swab your nose, you take a soil sample, you take something from the air or maybe the ocean even, and from there, with biochemical magic, you throw it on one of these next generation, or I guess like 5th generation or whatever that we're on now, Novikov X sequencing machine. I want to give you a picture of context of how big these are, because we often see pictures of sequencers. We don't get an idea that it's the size of a refrigerator. So you have all these microbial genomes floating around, and on this machine, one particular kind of technology is going to shred these, and then off of this machine is going to come off tens of gigabytes to terabytes worth of sequencing data. From this point, you might want to ask a few different questions. So for example, we got all these raw metagenomic reads floating around the place, and we also have a bunch of reference data. That is, genomes that have been previously sequenced of microbes, of bacteria, of viruses, of archaea, etc. So you have all these genomes, and depending on what database you use, these can be hundreds of thousands to tens of millions in size. One natural question to ask is, okay, these reference genomes have been a little bit more well studied, annotated perhaps. What about a goal of figuring out the presence or absence of one of these reference genomes in your metagenomic data? So in essence, we want to go one by one through these million or so reference genomes and ask, did I see it in my sample, or did I not see it in my sample? So essentially, abstracting this way away, we just want a binary vector, presence, absence of these reference genomes in my sample. One big problem that plagues metagenomics is that how do you disentangle noise from low abundant organisms? So if you look at like a typical environment's distribution of organisms versus their relative abundance, they often have roughly this kind of shaped curve. Way down here with these low abundance organisms, it's often hard to figure out, are these actually present? Or is this noise introduced by whatever computational method that I'm using to solve this problem? Unfortunately, current approaches are somewhat ad hoc. So you will see in publications oftentimes hard thresholding. You know what, we're just going to ignore all organisms below 0.01% relative abundance. Or some uninformed threshold of we're just going to make a cutoff, et cetera. And unfortunately, false positives remain. So this ignore all this low tail in actual data can lead to things that look like this. So I guess this is simulated data, however. In the blue here is the ground truth of the relative abundance of the organisms in the sample. And then Kraken, and its next generation Bracken, which is one of the more popular tools, is shown here in red. And so you see, in this case, this long tail of low relative abundance is indeed all just noise that is being introduced by this computational tool here. All these things aren't actually present in your sample. But it's still hard to figure out, should I set off my threshold here, or should I set it here? And how do I know it when my environment has a slightly different distribution? And this has also been seen in benchmark studies. So the critical assessment for metagenome interpretation looks at a bunch of different tools and looks at taxonomic profiling, finding the presence and relative abundance of these organisms in a sample. And in red here is shown precision. And you can see some well-known popular tools like Kraken via Bracken has precision that's essentially 0. So it lights up your entire database. It says, yep, your entire reference database is present in the sample. And then it's up to you to set a threshold to figure out what you should throw out or not. Okay, so that was the motivation for trying to develop YACHT to be able to do this thresholding in a little bit more of an informed fashion. It relies critically on this concept of average nucleotide identity or aligned nucleotide identity. So in case you don't know what it is, a quick example. It's going to be the ratio of matching versus all characters in an aligned pair of sequences or DNA sequences. So here's an example. I have 2 sequences. And notice that the red highlights where they differ. I have 15 nucleotides total. And between these 2 sequences, in 2 places they don't agree. So in this case, the ANI is 13/5. So if you have an ANI of 1, that says it's identical in the aligned regions. ANI of 0, they're as completely unrelated as possible. And this is very useful for practitioners because oftentimes we can find important taxonomic and phylogenetic differences based off of ANI. So here in this plot, I have ANI on the x-axis and then count of different comparisons between organisms of the same species or different species. And this is quite handy because if you have 2 things and you haven't characterized them, but you know their ANI is like 0.99, you can say, yeah, they're definitely the same species, even if you don't know what they are. So it's a great use for practitioners. Okay, so now that we have that in mind, what are some observations about the actual problem that we're trying to solve? So first off, no organism in a metagenome is going to exactly match something in your reference database. Bacteria in particular, and viruses, evolve quite rapidly, and also due to sequencing error, you're never going to get an exact match of something in your reference genome. So if you have a reference genome in your metagenome, and you look at the average nucleotide identity between one of these reference genomes and something in your sample, if you say, oh, it looks like it's 97% ANI related to something in my sample, does that mean it's actually in your sample? Or it's just related to something that's in your sample? What if the ANI was 92%? Is it in your sample, or is it just related to something in your sample, et cetera? Similarly, we have some problems about coverage. Because of this sort of exponential-ish distribution, all the organisms in there are not guaranteed to have even 1 times coverage. So what do we mean by coverage? Well, here in the black bar represents the genome, and the blue represents the different reads. And here I have at least 1 read covering every base pair, so 1x coverage. But those low abundant organisms often have lower coverage than 1. So in this case, what fraction of the genome do I have to see to be confident that it's actually present? Okay, so the desire for this method that I'm going to present is a statistical test based off of average nucleotide identity to something in the reference and incorporating coverage, say whether it's in or out of your sample. So at a very high level, how YACHT works, and I'll go into more of the algorithmic details later on, but essentially you give a confidence value and a desired false negative rate. And this will be important. It's a false negative rate, not a false positive rate. And you give an ANI percentage that you think means the same genome. So if you're doing strain level analysis, you might say 99.5% ANI or higher. If you're doing species level analysis, maybe only 95% ANI. If you're like, yeah, I don't care, I just want to get the phylum correct, maybe 70, 80% ANI. So depending on your application, you get to set this. You also get to set what coverage you think is enough evidence to support one of these reference genomes being inside the sample. And then YACHT, this approach, will tell you, is there enough evidence in terms of overlapping k-mers to support the claim that this reference genome is present in your sample up to within whatever ANI threshold that you set. So in essence, we no longer have to do hard thresholding. What we can do is we can go individual organism by organism or taxa by taxa and say, do I see enough evidence for it being present or not? The algorithm itself, don't read this, but just count the number of line numbers. It's 9 lines long. It's a very short algorithm, but it uses a lot of tricks in the background, and the bulk of this talk is going to be talking about those little tricks. And here's that little hypothesis test that I'll talk about. Okay, what are the tricks? Here's one of them. It's based on a sketching approach. So first off, how many people here use sketching, hashing-ish based? A few, couple. Okay, definitely not the majority. So quick introduction about sketching. Sketching is a way to do dimension reduction and preserve distances. So imagine you have a picture like this one, has a whole bunch of data in it, right? Very high resolution, multiple color channels, et cetera, et cetera. But to get sort of a basic idea of what's going on here, you only really need the skyline, which stores a whole heck of a lot less information. And the idea is that our sketch, literally a sketch here, similarity and differences between different sketches is going to be a proxy of similarity and difference of the high dimensional data. So in our application, we have a whole bunch of reads. And the sketching method that I'm going to use is called FracMinHash sketch. And it's going to extract a subset of k-mers from these reads. And then if I take another read set, could come from a metagenome or maybe another genome, I sketch it, I get another set of k-mers or hash values associated with those k-mers. And again, measurements between the sketch is going to be a proxy of measurements between the underlying read sets. Quick note, that's important. I'm talking about k-mers here in metagenomics. We're calling the k-mers, substrings of length k, like this for formers. But FracMinHash is just a general sketching technique. It doesn't care what the input data is. It doesn't care that their DNA sequences are k-mers or whatever. It's general purpose, and so you can use it for a lot of different applications. Awesome. So intuition behind FracMinHash sketch, you take the reads, you convert these into a set of k-mers, here represented by a bunch of dots. FracMinHash is going to subselect from these k-mers up to a certain fraction of your entire data. So essentially you set some fraction between 0 and 1 of how much you want to randomly sample. You sample those, and then the associated k-mers become your sketch. You can do this with your other read set, and then here again, the overlap between the two different sketches is going to be a proxy of the similarity between your read sets. How does this thing actually work in practice? No questions so far? I'm flying through this. Yeah, thank you.
So you're basically converting like sentences to bags of words?
Precisely.
Words.
Yep. Yeah, so this was all motivated by Broder's MinHash paper, which was looking at exactly the same thing. His bags of words were websites and the words on them, and he wanted to do intelligent indexing, so don't index similar web pages, and he did that same bag of words trick. Yep. Okay, so for FracMinHash sketch, we have this scale value s between 0 and 1, and I have myself a hash function. This hash function here is going to map k-mers into an interval of numbers. Importantly, it's an interval of known length with a maximal value of h. And that's going to be doing my random sampling in the following way. Oh, by the way, why is it called FracMinHash sketch? Really interestingly, independently, a lot of different researchers all came up with this same idea, mainly because the field had recognized that just MinHash sketches aren't useful in a few different applications. And they were called a bunch of different things. min code sync mers, FracMinHash sketches, variable minhash sketches, things like this. And it turns out that all of us got on Twitter and sort of like hashed it out what we're all going to call it to set it the same. So FracMinHash sketch right here is going to be all of those hash values who are some fraction below the maximum after you stick in all the k-mers in your set. So a pictorial representation of this formula, we have the set of k-mers, our hash values below some fraction. Imagine your k-mers sitting around, and I have my number line right here from 0 to 100, and 100 is going to be that h there, my maximum value of my hash function. And then I'm going to take this hash function, evaluate on each one of the k-mers after setting a threshold here set to 20%. So I take the k-mer, hashes it to a random number, take another k-mer, hashes it to a random number, repeat, repeat, repeat. After you're done doing this, you only retain the hash values or the k-mers that fell below that threshold. So that becomes your sketch. And so you can see how this allows you variable level compression, because if you wanted to, you could have set this threshold higher or lower, which makes your sketch bigger or smaller, which conversely, provably via formulas and things like that, allows your similarity approximations to be better or more approximate. After looking at FracMinHash sketch, there's been an explosion of different things that you can do with it. So I wanted to give you sort of a real brief tour of that here. We can calculate average nucleotide identity with it. You can also calculate other metrics like cosine similarity using these FracMinHash sketches. You can do taxonomic profiling, which is essentially what I'm talking about here for metagenomes. You can do functional profiling here. You can also do things like DNDS estimation. DNDS estimation is a ratio of the non-synonymous mutations versus synonymous mutations and allows you to characterize a positive, neutral, or negative selection. And community has grown around this, so there's a lot of user-friendly tooling to be able to use these FracMinHash sketches. So one other interesting connection that I wanted to do with the talk that I gave during the retreat is that these taxonomic and functional profiles that you can get out of using these FracMinHash sketches, we have a way that you can integrate them into a knowledge graph if you want to, which connects these predictions of taxa and function to things like gene ontologies, antimicrobial resistant genes, diseases, phenotypes, stuff like that, for doing different things like hypothesis generation, pathogen prediction, machine learning stuff with embeddings, or just straight out exploration. So, side note, FracMinHash has seen lots of usage recently. Great. Okay. So, how does it work? Two key recent developments allowed this YACHT approach to be developed. First off, relatively recent results relating average nucleotide identity, equivalently mutation rates, to different k-mer-based quantities. So please don't read these formulas, but essentially what we're able to do is relate the number of k-mers that either have a mutation or don't to things like a mutation rate or average nucleotide identity. And then similarly, we were able to show that these FracMinHash sketches, with a slight factor that's added in there, can give you an unbiased estimate of ANI. So essentially, your FracMinHash estimate of similarity, here called the containment index, is related to your ANI value. So with those two things in hand, we can now see how does this YACHT algorithm actually work. Recall, what we have, a bunch of reads from a metagenome, and we got ourselves a reference database of 1 million or so different microbes. Very first thing we're going to do, since we're taking the bag of words approach, is turn these all into k-mers. So I'm going to turn these all into k-mers, and then after that, I'm going to subsample or sketch, here represented by those little red dots all over the place. Next, there's no reason to consider any of the reference organisms that don't have anything in common, i.e. no overlap of k-mers, with your sample. So we're going to only retain those with non-zero overlap. After that, we're going to take a little cue from Kraken / Bracken / a lot of these other cases, and we're going to look at informative k-mers, that is those k-mers that appear in a single reference genome and not other ones. Here, we have a lot more of them because I don't do it for the entire reference database, just the ones that have non-zero overlap with my sample. So then we have to go and calculate if there's enough support for the reference genome being in the sample. That was this hypothesis test that I was talking about, and I've mentioned it twice now, so how about we go ahead and discuss what that is. What is this hypothesis test? Well, just like from Jessica's talk yesterday, she talked about Bayesian versus frequentist. This is a very simple frequentist approach. Our null hypothesis here is a little weird. I actually took a bit of back and forth with the referees to be able to get them to understand that the null hypothesis here is that the genome, the reference genome, is present in your sample. And we'll see why we can do that, or why that's critical, in a second. So we have our genome. Remember, I split it up into a bag of words, in this case, k-mers. And I'm assuming, for my null hypothesis, that all these k-mers are present in the sample. So every single one of these blue bars in my genome present in the sample. But however, remember I said that none of the reference genomes are exactly going to be represented in your sample. They're always going to be off by some average nucleotide identity. So that means that we have some mutations in our reference genome in comparison to what lives in my sample. Here represented by that little red dot there, I guess right here. And that affects a bunch of k-mers. The k-mer size that I'm using is relatively large, like 21 to 51. So with very high probability, if you introduce a mutation here, this k-mer is not going to show up in your sample, as indicated by the absence of all these red ones in my sample. Okay, and now here's one of the critical parts of the sketching. After I do this, because here I'm talking about all k-mers, if I now subsample this with a FracMinHash sketch, notice that, again with high probability, my k-mers aren't overlapping. So that means information about one of these k-mers being mutated doesn't affect any information about another one of the k-mers being mutated. Therefore, the number of k-mers that are in common with my sample, that is all these blue ones, is actually distributed according to a binomial distribution. For every single one of the k-mers, I just look at every single one of the base pairs and say, what's the probability of it mutating? Or I guess, what's the probability of it not mutating? Times, go to the next base pair, what's the probability of it not mutating, et cetera, all k times, and we get the probability of success of a k-mer not having any mutations in it. And we do this for every single one of the k-mers in my sketched reference genome. So then, if I take a random variable that's distributed binomially with these same parameters here, and I take an integer that's large enough such that the probability of this random variable being bigger than it is greater than this alpha here, which represents our significance, which is related by 1 minus to our false negative rate. Okay, there might be a lot of probability, but essentially what this says is this tells you the fewest k-mers that we would see with probability alpha if the mutated genome was present in our sample. So then you can go and say, well, for my actual reference genome, what do I see in terms of overlap? If you go with your reference genome, intersect it with your sample, and you see that there are enough of these k-mers in common, you can say, aha, it's in the sample, I can accept my null hypothesis. Otherwise, if you don't see enough k-mers, you can reject this null hypothesis. I'll pause here. Okay. So how good is this test? To be able to characterize that, we would need a statistical power. So we're controlling our false negative rate, as I indicated right here. 1 minus this false negative rate is this alpha. So what about false positive rate? So unfortunately, to characterize that, you would need some distribution of known average nucleotide identities. So think about it. If you have a reference genome, and it is 0% ANI to your sample, it's real easy. You can be very high-powered and confident that no, it's not present. So you'd need a distribution over these values, and we don't want to do that. We want to do this distribution-free, because similarity and differences of organisms in a metagenome is very much environment dependent. So instead, we calculate this other quantity, which is how small must the ANI be until my false positive rate is the same as my false negative rate. In other words, how distantly related must things in my reference in my genome be for me to correctly say, nope, they're not present, so that I don't get these false positives. So again, don't read this formula here. Let's look at a graph instead. So on the x-axis, I have average nucleotide identity, and on the y-axis, I have the positive rate. And so notice, if my reference genome over here is a very high average nucleotide identity, then I correctly detect that, yes, this organism is present in my sample. If my reference genome is distantly related to my sample over here, I correctly reject and say, no, this reference genome is not in my sample. And then we have this really wishy-washy area right here in the middle, where sometimes I say that, yes, it's in, sometimes I say, no, it's not. But if you look carefully at these curves here, this blue curve and this orangish yellow curve right here, this corresponds to the number of different k-mers I'm talking about. So if you imagine, if you increase more and more and more k-mers, this plot is eventually going to look like this nice whoop, spike up like this. And then exactly at whatever ANI threshold you set, your concept of organism identity, you're correctly saying that it's in or correctly rejecting that it's out. So look in the appendix for some nice proofs about this. Okay, how about experiments? So there are different knobs that you can twiddle for this approach. You can play with the k-mer size. And very interestingly, we have some results about how the power is non-monotonically related to the k-mer size, so that there's this sweet spot between k-mer size that maximizes your power. You also have the ANI threshold that I've been talking about on and on, what constitutes the same genome, that significance, your false negative rate, and your minimum coverage. When I gave you the formulas for how the hypothesis test worked, I didn't talk about coverage, but basically it's just divide by coverage and you're good. We find that this min coverage and ANI threshold are the two most important knobs to twiddle, so I'll talk about those first for my experiments. So first set of experiments, let's do some real spike-in data. We take a real metagenome, so this real metagenome, not mock, I have no idea what's in it. And then I take a real genome, and I'm going to artificially mutate this thing and stick it in my sample. And now I have this ANI threshold. So I can mutate it so that the ANI is large. Actually, let's go ahead and look at this plot right here. If I do a small mutation rate, then it's highly related to my sample. It's above this ANI threshold. I am correctly saying that, yes, this organism is in my sample. Similar organism detected is true up here. If I mutate this a whole heck of a lot, my ANI is going to be low. And so I'm going to correctly say that, no, it's not in my sample. And then I have some false positives up here. It's really hard to see because there's about 30,000 dots on this plot. But this false positives up here represent just under 3%, and this over here under 1%. 1% was the desired false positive rate that I set. It also behaves as expected. So if you consider this plot here, where on the x-axis I have that minimum coverage, what percentage of the reference genome's k-mers do you have to see to say, yep, that's in my sample? Versus your false negative rate. If you say, I only need to see 0.1% of the reference in my sample to say that it's present, of course your false negative rate is going to be real low. Similarly, if you need to see every single one of its k-mers presence, your false negative rate is going to increase. And on the flip side, same with the false positive rate. If you only need to see a little bit, you're going to get a high false positive rate, and conversely with a higher coverage. So in general, we find about 5% or higher is probably a safe bet. Okay, so what about benchmark data? So I mentioned before this critical assessment of metagenome interpretation, that's abbreviated CAMI. So there's a bunch of benchmark data set. I used it from the second round of the CAMI experiments. Here is the Rhizosphere challenge, which is one of the most difficult benchmark data sets. The way the competition worked is they gave you the data set, it's simulated, they don't tell you what's in it. It actually has genomes that have been referenced or sequenced but not put into reference databases yet, which is cool. But in any case, you can see in color some other methods that do this approach, and then in this grayscale, our YACHT approach. I've connected these with a line right here just for aiding your eye to see that this talks about different coverage rates. And you can see indeed, as you increase your coverage up to 1, your precision or purity increases, and conversely, if you make it smaller. But the takeaway here, a perfect method would be all the way up at the top, and we have our other methods here. That was the most, I guess, beneficial for YACHT comparison. A sample that most tools do relatively well on, this marine sample here, looks at a bunch of other tools, and again, the YACHT results are highlighted here in this grayscale. So by changing this minimum coverage, you get a tunable parameter for false positives and false negatives. Interesting future work could be, what's the top rightmost setting that you could use? And similarly with the strain madness data set that looks at a whole bunch of strains, here again, tunable and outperforming some methods, not all, but some in certain metrics. Great. I anticipated more questions than this, so I think I'll be able to get us back on time because I want to talk just now in the conclusion about, oh, yes, question, yay.
Is there a way to control for different species having different mutation rates, so they're not going to have the same level of difference from the original reference?
Yeah, great question. So in certain clades in a phylogeny, you're right, the evolutionary relatedness within that clade varies wildly across the tree of life. Yeah, so the cool thing here is this test is a 1-by-1 test. So what you could do is you could run YACHT on a certain clade that, let's say, they're highly related with an ANI threshold of 99.5. And then when you step over to this other clade and you want to say, are these present or absent? You notice that they're very distantly related. And you can get away with a 90% ANI. You could then run the test there. So you don't need to do it on the entire tree of life at once. You just take one clade, run it, take another clade, and run it. Yeah, great question. Okay, features, future, friends, and funding. This is published in Bioinformatics. The preprint is QR code linked right there. And the code is also on GitHub. We've been trying to up our game in software quality. So let's see if I can show you over here. Where's my mouse? So for the first time, we actually have unit and integration tests and code coverage and all this kind of good stuff to try to make it like robust software that you can use, available on Conda and all your favorite other channels like that, and including things like wikis giving brief descriptions, tutorials, pre-trained data sets that you can download, and you don't have to download these automatically. You can actually do it, or sorry, you don't have to download these manually. There are ways that we can do automatic download of reference databases and things like this. So try to do a lot of end user creature comforts in this implementation. If you find a problem with it, please do submit an issue or a pull request if you find a solution. Okay, so in conclusion, oddly enough, as far as I'm aware, I've been doing metagenomics for quite a while, this is the first statistical test for presence absence of a metagenome, of a genome in a metagenome. Usually before this is these ad hoc thresholds that I was talking about. It obviates the needs for those arbitrary thresholds, and then hopefully these are intuitive parameters that practitioners and biologists and bioinformaticians can understand, ANI and coverage. And one thing that I didn't talk about here is it's very scalable. Since this is a sketching-based approach here, it's ridiculously fast, especially since you can control how fast and accurate you are by how deeply you sample with those FracMinHash sketches. So what I want to do now, or next, is we have collaborators and other people who have done FracMinHash sketches of all metagenomes in the sequence read archive. What I want to do now is chuck it at YACHT to do functional and taxonomic profiling, put all of these on that metagenomics knowledge graph that I talked about to do an interesting integrative study. I'd like to thank Stephen White, a mathematician, PhD student who's recently graduated, Chun-Yu Ma, bioinformatics and genomics PhD student who's also graduated on the job market. Collaborator, colleague in the math department, and a part of the Koslicki lab who helped with writing the software, making it easier to use and intuitive, et cetera. So thank you for your attention. Oh yeah, and funding, of course, in IHR1. Without them, couldn't do anything. Thank you.